아키아에 대해서 실무에서 많은 경험을 갖고 계신 분이 이제 론이라는 분이 세미나를 하시는데요. 지금 국내에서 이 아키아에 대해서 잘하는 분이 별로 없습니다. 저도 잘 모르고 있고. 그래서 이것이 아마 이제 최초의 아키아에 대한 세미나가 아닌가 싶은데요. 지금 특히 이제 사우스 아프리카를 위주로 해서 현장에 적용됐고 그런 예를 지금 여기서 보여드릴 거니까 이걸 보고 우리나라의 어떤 방향으로 이걸 적용을 하고 이용할지 생각해 볼 수 있는 계기가 됐으면 좋겠습니다. 오늘 말해주실 분은 로니 엠바스라고 하시는 분인데요. 거의 물 관련된 특히 이제 바이오로지컬 웨스트 분야에 15년 이상 경험을 갖고 계신 엔지니어입니다. 특히 이제 이쪽에 아키아를 이용해서 실질적으로 핫스토리장을 업그레이드 시키거나 효율 향상시키는 공정의 일을 많이 했기 때문에 아마 오늘 아마 저도 영어를 잘 못하기 때문에 제가 통역이 좀 잘 안 되고요. 천천히 하시면 아마 대부분 알아들으실 수 있을 거라고 생각을 하니까 천천히 하도록 부탁을 드리고 세미나를 시작하도록 하겠습니다. 그리고 나중에 질문은 여기 영어 잘하시는 분들이 김 선생님도 계시니까 통역이 충분히 되려고 있고요. 질문하실 거 있으면 메모해 주셨다가 끝나고 나서 질문을 해주셨으면 좋겠습니다. Okay, now running your turn. Everybody here maybe can be understand English, but if you speak slowly and clearly, no problem here in English.
Thank you.
Can you show the slides?
Professor Tom, thank you very much for the introduction. Gentlemen, thank you very much all for coming. I realize there is a language barrier, so please at any time interrupt me and I will explain, or maybe Professor John or Mr. King will be able to translate. I will be talking on how to enhance your sewage treatment works. How do you get the most out of your sewage treatment works? And the way we see it is by using Archaea. Archaea is the key to wastewater treatment. And how does it all fit into the system? If you're looking at wastewater treatment plants, what is a sewage treatment plant? Most people will tell you it's an activated sludge basin, it's a lot of concrete, there's aerators, there is electricity and all the rest of it. But if you look at a biological process, the essence and the focus of a process is not the concrete, it's not electrics, it's not the mechanicals, it's the biomass. How do you get the biomass to function optimally? How do you make them work harder? So how does it work in nature? Nature was always been composed of two components. You had your eukaryotes, which was basically your algae and your fungus, and you had your bacteria. And everything was basically divided into those two domains. Humans, just for the sake of interest, they fall on the eukaryotes. And about in the 1980s, there was a gentleman by the name of Carl Weiss. And he basically discovered a third life form. And the reason why he only discovered it was we didn't have the equipment to really see or find them, discover them. They've always been there for millions of years. But there were certain environments where that were always considered as being sterile, like at the sea bottom floor where you had hydrothermal vents, under 350 degrees Celsius, you're looking at 7-8% salt concentration, maybe even higher. Very high in sulfur, very high temperatures, low pHs, and they've now discovered that it is actually teeming with life. But not life as we know it. It's not bacteria, it's not plant and animals, it is archaea living there. And they found that these archaea, as they are now looking more and more into marshes, into everywhere, they are finding more and more of these archaea established even in oxygenated areas. Alright? So in the absence of any of these species, your recycling of carbon, nitrogen, phosphorus, whatever, will cease. And if archaea were not present, we would have been up to eyeballs into waste. So the natural way, how did nature always degrade wastes? And this is how I see it. If you're looking at the simplest way of waste sewage works, you're looking at an oxidation pond system, where you have organics coming in, your biological material settling to the bottom, you have your organic waste being degraded into methane, ammonia, and all that, CO2. And they actually become a food source to your bacteria. They produce CO2 in turn, and in the oxygenated top zone you have your algae consuming the CO2, providing the oxygen to the bacteria. When these organisms die, they settle to the bottom, and they in turn are then decomposed into methane, CO2, and the whole cycle continues. You have sunlight, you have wind introducing oxygen and CO2, and this system is in balance. And if you have, in our country, South Africa, we have lots of oxidation pond systems, and if they are designed properly and correctly, on correct loadings, they will function properly for years, for even 100 years, without a problem. But especially in Korea, there is a problem with space. Everything has to be compressed, everything has to be optimized, there are too many people. So man has done away with this kind of setup, and what they've done is they've replaced the oxidation pond system, put it into an activated sludge system, and they've introduced oxygen by using aerators instead of the algae. If we're looking at anaerobic fermentation, you're looking at the glucose is converted to acetate. Alright, that's the first step, acid formation. However, the byproduct is H2 formation, and obviously your pH drops. And if you don't remove your hydrogen, you have a problem with anaerobic fermentation. So what happens if you have a buildup of hydrogen and you cannot remove it quick enough? You are starting to produce other higher order acids, propionic, butyric acids, and they are more difficult to break down. The only way to really make this system function properly, and organisms prefer to break down glucose to acetate because it's more energetically favorable for them. They get the most energy out of it. And the guys that do that work is Archaea. The methanogens have always been classified as being bacteria. They are not bacteria. They are Archaea. They've been reclassified. The interesting thing is, if you're looking at an anaerobic digester, the population of Archaea in the digester is only 4% of the total. The rest of it is all bacteria. The only thing bacteria do is converting your glucose to acetate. In other words, converting it, not converting the COD to anything. You're changing the structure from glucose to acetate. So you haven't removed the COD. You've done shorten the chain, and you've produced a lot of acids. And the only way to get rid of these acids and to remove your COD is to let the Archaea remove the acids. And if you don't, you haven't removed any COD. The only way to remove COD is to methane. So Archaea, what they do is they regulate the mechanism of acid-forming bacteria by shifting the fermentation reaction towards the formation of acetate. And what I basically mean is the faster you can remove your hydrogen, the faster that reaction will move to the right. So if you have a restriction on your hydrogen removal, you have a problem. You will not be able to remove all your COD. You will have, let's say, 80% removal, and the rest will be either the acetate or some of these other acid forms. Whereas if you remove your hydrogen fast enough, 90, maybe 95% will be removed to methane, and the rest will be some of these other acids. So that is how, by adding and introducing Archaea into anaerobic digesters, for instance, you can alleviate the stress out of the system, and you can actually increase gas production and obviously the feed rate. And this is some of the results we obtained. This is a study that was done in the States, Mexico State University, where they took dairy waste, one control, one treated, over a period of 720 hours. The gas production on the top is the Archaea system, and the bottom one is the control. Same conditions, same feed, same everything. And as you can see, the percentage increase versus the control is more than 100%. More than 100% gas production. So, increased gas production, which means an increase in metabolism, which demonstrates the movement through from glucose to acetate to methane. Get it out of the system. There's no stopping it. And that's what the Archaea do. Increased rate of sludge utilization rate, stabilization, so the rate at which you can pump through an anaerobic digester is increased. Your feed rate is increased. Normally a digester can take X amount of sludge to digest it. And if you're looking at these numbers, we can almost double the feed rate and still get the same results. The other thing as well, it reduces odor. Coming back to the Archaea, there are basically three groups. You get your methanogens, then you get your Extremophiles. Extremophiles mean they love extreme conditions, like low pHs. They've been measured to operate at below pH 1. As I mentioned, 350 degrees. They live in high saline conditions as well, in salt pans they love to live. And then you get a group as well, they're actually removing the sulfur. Their food source is sulfur. They do not accumulate it as sulfur in their cell structures. They actually incorporate it in their cells. So it's not a granule inside, it's actually part of their cell structures. Dr. Charles can embroider more on that. If we're looking at aerobic respiration, like we have in an activated sludge system, your bacteria convert glucose to CO2 and water. That's how you're removing your COD. And you require oxygen to do that, and bacteria do that. However, when you have a problem with providing oxygen in your system is under stress. And it does a metabolic spillover, overflow. And what happens, the glucose is not converted to CO2. It's converted to acetate, formate, ethanol, and in that order. The reason for that is depending on the amount of stress the system has. So what happens is, these byproducts you can call them byproducts that are produced. They slow the bacteria's growth rate. These bacteria are not growing optimally because of these byproducts being produced, but these byproducts are ideal food sources for Archaea. And they're converting the acids into methane, CO2, and water. Now, how does it work in an activated sludge system? If you consider an activated sludge system, we have biological sludge, which is floating around, it's being mixed by your aerators, oxygen is produced, and when your oxygen concentration, typical in activated sludge system, is above 2 or maybe higher, it penetrates right into the biological floc. However, when your DO concentration drops, it does not penetrate completely. What happens is you get an anaerobic core. It only goes up to there. And let's say from this state onwards, you are producing all these metabolic byproducts. And what happens is the Archaea establish on the inside. So you have a nice family of organisms and they help each other. It's a synergistic effect. So one of the impacts, some of the studies and work we've done is they're removing the metabolic byproducts that typically inhibit bacteria. Okay, this is the control curve, the bottom one. This is the endogenous respiration rate. This is a sequential, was done on a sequential batch reactor. Oxygen utilization rate, I hope you're familiar with that. So initially when you feed a sequential batch reactor, your oxygen utilization rate shoots up. And this point basically tells you all the readily biodegradable material is consumed. And then you get a slower slope when all the particulate COD has to be broken down and metabolized before it's consumed. That's why you have a slower, lower slope. And then you get a dip, and then you get your nitrification. When we dose the system with Archaea, the interesting thing is the endogenous respiration rate goes up. We have a much higher peak. Your oxygen utilization rate is a lot higher. Some of the interesting things as well is, if you look at this slope, the slope tells you your reaction rate is a lot higher. The rate of substrate consumption is a lot higher, and you reach endogenous respiration a lot quicker, about, in this case, more than 30%, which means you can do the same job in a smaller reactor. So let's work through those points again. The increase in biomass activity and viability, that's what we can read off this curve, the oxygen utilization rate, tells you we have an increase in biomass activity and viability, is more active. The slope tells you we have a higher mineralization rate of your particulate matter, and the increased rate of oxygen utilization rate. The interesting thing which I forgot to mention is the bottom curve, the control curve, was done at a dissolved oxygen concentration of between 1 and 3 milligrams per liter, which is typical of a system. Ours was operated, it's actually 2 and 5, sorry, and ours was operated between 0.5 and 1.5, which is typically we would consider as being stressed. Anything under 2 milligrams per liter dissolved oxygen, you have a problem, especially with nitrification. Okay, and then the shorter cycle time, so we can do the same job in a smaller reactor. This is the performance curve we got out of little pilot plant we did with Sasol. Sasol is a big company in South Africa, petrochemical company. where they start off with a COD of approximately 20 to 30,000, and they had a typical performance of about 85 up to 90%. Interestingly enough, they added micronutrients before we started. So the average performance is less than 85%, which is normal if you consider you're trying to break it down from 20,000 to as low as possible. This COD was about 4,000 or 5,000 milligrams per liter. COD as chrome. We only use chrome. Okay? As soon as the Archaea were introduced, the performance increases to more than 95%. Fortunately, the system was stressed. It did not have sufficient oxygen. The sludge yield decreased. The sludge yield being the kilogram of sludge produced per kilogram of COD removed. And it dropped from about 0.2 to 0.05, which was a close to 70% reduction in sludge production. And the simple reason for that is if a system is under stress, you're producing the acids. The acids are food for Archaea under low dissolved oxygen concentrations, and they are removed, in other words, as methane, and you don't produce excess biomass. We then did a full-scale trial on the same wastewater, and we had exactly the same results. The COD came down dramatically from about 5,000 to, I think in the end we dropped it to less than 2,000. The suspended solids, one of their concerns, main problems they have is suspended solids carried out of the activated sludge reactor. We lowered that from about 3,000 to 400. And the sludge production was reduced once again from about 0.2 to 0.05, basically confirming what we've achieved in the lab. This is rather interesting. An activated sludge system, if you do the numbers, in the way I do it is the modified or UCT model. You can calculate based on the volumes you want to treat, the amount of COD you have to remove, how big a reactor you need. That's a given for a certain sludge age. So you can draw up these curves, and this was done for a reactor that is supposed to treat 20 megaliters a day, which is 20 tons, more than 20,000 tons, and then operated at certain mixed liquor concentrations. This was the straight line is the actual basin size. Okay, it's less than 2,000 cubic meters, less than 2 megaliters. So the residence time in this basin is approximately 2 hours. So if you want any nitrification to happen, you would require to run extremely high mixed liquor concentrations. As you can see, to get underneath this straight line, you need to run at about 6,500 mixed liquor total solids concentration. But that will end you or give you only a 10-day sludge age. And if you want any nitrification to happen, you would require it to be almost at 15-day sludge age. That's straight theory. They, on the other hand, were running the plant at 3,500 milligrams per liter, and they were not achieving any nitrification. So we were asked, could we achieve nitrification on that plant? And yes, we did. This is typically the ammonia out of their reactor, which is about 20 milligrams per liter. As it comes in, it goes straight out, no nitrification. And as soon as we started, as you can see, the Archaea are established in the system, and here ammonia concentration drops. Similarly, COD concentration is improved. And the bottom one is suspended solids. We dropped that to less than 10, which is basically negligible amount of suspended solids carried out of your clarifier. once again, we had to convince them to reduce their sludge wasting rate to allow us to contain a lot of mixed liquor in the system. And so once again, we confirmed what we saw at the petrochemical plant, reducing sludge production by at least 50%, dropping it from 0.3 approximately to less than 0.2. That means you have 50% less sludge you would have to dispose of. Biofuel processes work on the same principle. The biofuel processes have this advantage that you automatically have an anaerobic section in your biofuel, because oxygen only penetrates about 150, maximum 300 micron. So if your film thickness is a 500 micron, maybe 1 millimeter, the rest is all anaerobic. So your substrate goes all the way in, but not your oxygen. And in a biofuel system, the main problem is always its oxygen. Looking at biofilters, this is the trickling filter. You saw the photograph of. The bottom curve is typical what you will find in the literature, and it's been confirmed on numerous studies, that if you increase your organic loading to a trickling filter, your removal goes down. If you want about a 90% removal, your loading should not be higher than about 250 grams of COD per cubic meter. As you increase the loading, it slopes down to approximately 50% COD removal. The rest just goes out. It cannot consume that. You don't have enough oxygen. Whereas in an Archaea system, we have a 90% removal all the way from 250 all the way to 1.5 kilos of COD per cubic meter. That is substantial. That means you've increased the capability of your trickling filter by 500%. Looking at nitrification, nitrification is the same. What happens is, if you want complete nitrification, you would need to load your system about 200-250 grams per cubic meter. You want to remove sufficient ammonia. As you increase your loading, your nitrification drops to virtually zero, because they cannot compete for oxygen with the COD consumers. Their growth rate is too slow. With Archaea, we are way above what the theory predicts we should be able to get. We get about 50-60% ammonia removal out of this, under these kind of conditions, all the way up to 1.5 kilograms of COD per cubic meter. And the interesting thing is, as well, is we get simultaneous denitrification. In other words, the faster the ammonia is converted into nitrate, there is still enough space down that the nitrate is also consumed. We get 90% odd denitrification. We have a phenomenal amount of reduced sludge production, and it's been quantified as 70%, 70, humus sludge. That is humus sludge which you don't have to process through an anaerobic digester. It's sludge you don't have to throw into a drying bed or have to incinerate, as you have to do. If you're looking at, it's not very visible unfortunately, activated sludge systems, we've discussed. The plant is designed for about 15 megaliters a day. It takes 20 megaliters a day, and at 15 megaliters it was not able to remove any ammonia. At 20 megaliters, we're converting the ammonia by at least 50%, from 21 down to 7, it's actually more, 60-70%. The same on the trickling filters, and the same for oxidation ponds. You don't have any oxidation ponds in Korea?
No, I don't think so.
No, it's a pity. Okay, but anyway. if we're looking at what can we do, what does Archaea do for your sewage treatment works? You upgrade your process capability. One of the problems, you have to look at the alternatives. How do you upgrade your process capability? Normally, if your flow has increased or your organic loading has increased, what will happen is consultants will tell you you have to double your process capability. You have to build more concrete. You will have to put in more aeration, more sludge handling facilities. We say no, it's not necessary. By incorporating Archaea in your process, you will be able to achieve similar or even better effluent qualities with all the benefits of all these other things. It's less susceptible to shock loads. Our systems, because you are introducing Archaea all the time, it is more stable. Even activated sludge, it can take shocks. It is not, it's very tolerant to hydraulic or organic shocks. The effluent treatability is increased. We have got a plant which gets coffee effluent. Coffee, as you know, contains a lot of tannins. Tannins are extremely difficult to degrade. The plant, before we went on it, the effluent looked like coffee. And with the Archaea, it breaks down the coffee color. And it's actually clear, crystal clear effluent. You are decreasing your sludge production at least 50%, 50%. Your oxygen demand decreases. In other words, if your plant is overloaded, you would require additional aeration to be able to cope with the organic demand, the oxygen demand. We say, if you introduce the Archaea, you don't need the capital investment of introducing or buying more aerators, putting in the electricity, the maintenance costs involved. Archaea will remove those metabolic byproducts to methane. And the additional benefit then obviously is the reduced sludge production. We also increase sludge settlability, even with the current clarifiers. Even if the flow rate increases to about maximum, the biomass stays inside, it's not washed out, and your foaming and bulking problems are also alleviated. If you're looking at your anaerobic digesters, we have more gas and less sludge, once again, reducing any downstream impacts. So in essence, you are increasing your nutrient removal, you are increasing or improving your effluent quality, and most importantly, you are reducing your treatment costs. And that's what most people are interested in. Sludge production alone can be about 50% of the operational cost of the treatment works. 50%. Now, if you have to now upgrade your plant, and you have to spend capital, I'm not quite sure how it works in Korea, but in South Africa, you go to the bank, you borrow money, they want interest. That's a lot of money. So there's basically savings all around. Where do we apply Archaea? Basically everywhere. Anything that's biodegradable, municipal sewage, brewing and distillery effluents, tanneries, dairies and food industry waste, anything that's all very high in CODs, abattoirs, paper and pulp industries, all those recalcitrant compounds, petrochemical as I discussed, landfill leachates, textile, and anything basically that is biodegradable. In nature, Archaea has always been there. In our man-made systems, we are trying to optimize on space, on energy, on costs. But in that process, we have sacrificed, we have lost the Archaea. They cannot establish under those conditions. There is too much oxygen. There's too many other organisms. They cannot outcompete them. However, when we create the conditions and we introduce the Archaea at a constant rate, that's when we have all these impacts and we improve system performance. And that's the end.
네, 뭐 질문을